Welcome everyone for the second session of the Topological Image Analysis Mini Symposium, which is part of SAM 2020 Conference of Mathematics of Data Science. I'm actually very privileged to introduce to you Lise Munch from Michigan State University. I personally benefited a lot from her work and from her amazing papers. So I think if I go again through the list of the publications and achievements she did and her amazing group at Michigan, I won't finish. So without further ado, stage is yours, Lise.
Awesome, thank you. And thank you, Ars, and everybody for the invitation, and thank you everybody for joining us virtually. This is really nice. It's great to see such a good turnout. So what I want to talk about today is how to use, or at least how we have been using, TDA for quantifying plant morphology. And so the particular project I'm going to talk about involves a lot of people, which is always going to be true of any really truly applied project. So I am one of many, so this is a collaboration with Dan Chitwood, who's at MSU in horticulture, and our student Eric Amaskita, and some other members of our groups, and some collaborators that were kind enough to share some data. All right, so let's talk about plants. Let's see if that, so plant morphology. So there is a fascinating world of trying to understand shape and structure in plants in a way that we can encode information that's useful for understanding the plants themselves, the genetics, the output, the yield, all sorts of things we might want to ask about our plants. This has been an extensive field of research in general, but a lot of the tools, if you go in and understand what they're really doing in terms of trying to quantify the plant morphology, the tools that they use are incredibly simple, trying to measure very complex structures. And so one of the exciting things about these collaborations is that we've had a chance to bring in topological data analysis methods in a way that can actually quantify some of the more subtle structure that's encoded in this information. So these are all examples of plants that have been scanned with an X-ray CT scanner. So the colors are all false, so do not believe any colors you see. But the idea is that when we work with X-ray CT scanners, we not only get sort of the outside global structure of the plant, but we can also look at interiors and differences in densities, all sorts of interesting things that we can play with. So TDA, so this is just sort of my life philosophy slide, which is, this is basically what we're trying to do with any sort of topological data analysis method. We have some form of input raw data, which we saw a bunch of talks this morning that involved everything from images to point clouds to moving swarms to you name it, we can find kind of a way to mess around with it. Once we have some sort of input raw data, the goal is usually to find some sort of summary that encodes the topological structure. So these can be from the complex to the very, very simple. So we saw examples this morning of persistence diagrams. I think some people, I don't know if we fully went into things like merge trees and mapper graphs and reeb graphs and other sorts of graphical structures. But there's lots of ways of essentially taking our input data, which is potentially very large and hard to deal with, and summarizing it in some smaller structure that at least encodes most of the important information we want, hopefully. If we've done this right, then it encodes something about the structure. And then because we're actually working with data analysis, we need to be able to take the topological summaries and do statistics and machine learning and prediction and things like that on it. So a lot of the end goal of these things is trying to take our topological summaries and do something with that. So we saw examples of that this morning with things like persistence landscapes. There's other methods with persistence images and things like that. Today I'm going to talk about a really simple version of this, which is really just the Euler characteristic, which is great and something you might have seen maybe if you've had a high school intro to math class that was just, hey, Euler characteristic, but we'll get there in a second. So the overarching goal here, as I see it, is TDA, we're trying to build summaries of the shape of data that are particularly nice, especially in the face of noise. So we should be able to compute them relatively easy. Or at the very least, I should be able to have a computer program do it for me. I should have methods for comparing them so that I can start talking about metrics and understanding how good a job I've done. I can provide guarantees that way. They should be robust in the face of noise. So if I see a little bit of noise in my system, it should not freak out my topological summary so much that it's completely useless. And it should be concise in the sense that if I have a gigantic input, say 3D scan of a plant, the output should not be so large that I can't handle it. It should be some sort of summary of what's going on. So to that end, what we're going to talk about today is a project we've been working on that involves Barley. All right, so Barley. What is Barley? So it's something you can go buy in the store if you want to. Barley is one of the world's most ancient crops. It's the fourth most important crop behind rice, wheat, and corn. It's been around for centuries, clearly. It's useful for everything from animal fodder to making different types of bread. People make soups with it. You can make beer with it. So I appreciate it. But so the idea here is that it's been around for a long time, and it started as like most plants, a wild specimen, this sort of elongated shape. And over the course of human history, while we domesticated the barley, we basically selected for traits that we wanted. Over time, what we ended up with was a structure that's a bit more smaller, but more spherical and more focused on the kernels. So in particular, one of the traits that was largely selected for was the number of rows. So there's two types of barley. Two classes of barley. So there is 2-row and 6-row barley. So it's a little bit easier to see in the picture over here. Oop, there we go. Over here in the bottom right. Essentially what's going on here is that you have sort of the main stem that goes up the middle. And in the 2-row barley, you have sort of 2 kernels that come off of the side, versus the 6-row barley, you have 6 that are arranged in irregular hexagon maybe. And so if you were going to be, say, planting crops to create food, you would definitely want to have 6-row barley instead of 2-row barley. And so there's going to be a lot of selection for that in the domestication. So the data set that we have access to is what's called the composite cross-2. So this is an experiment that actually started in the 1920s, which I think is nuts still. So in the 1920s, this is Harlan and Martini, picked up 28 founders, 28 different examples of barley from all different regions in the world. You can see these guys over here. And they took all of this barley and started crossing it and planting it in fields. And so as with all forms of long-running experiments, which are well beyond the scope of anything I think we see usually in the math community, maintenance of this experiment and maintenance of the data is a long-standing process that involves a lot of people. And so I'm lucky enough to have down here, this is Dan Koenig is our collaborator who has all of this data over 50 years worth of planting this barley. All right, so the basic design is the following. So we have 28 parents, so the 28 founders that we brought from the different regions. And these were taken and specifically crossed. We got all pairs crossed. So we have 379 hybrids. Each of those hybrids is then planted out in its own field and allowed to self-fertilize. So in theory, they all sort of fertilize with their own hybrid. There's going to be some cross-pollination, but for the most part, it tends to stay pretty stable. And they did this for 50 generations, so 50 years. And then now we have access to all of these seeds from 50 years worth of data, and we have examples of barley spikes and all sorts of different information in terms of shape and structure all the way down to the genetics that we can play with. And so I'll point out for those of you that are paying attention at home, 28 choose 2 is actually 378. There was an error somewhere in the original experiment, which is why we have 379. So nobody freak out in the chat. Okay, so this is our experimental setup. So we got access to the data. By access, I mean literally shipping spikes across the country, which is pretty cool. So the idea is that we have, so Dan Chitwood's lab has this X-ray CT scanner. So this is this gigantic box. This is actually a picture of it before they installed a room around it. They actually built the walls around it. That thing is just basically a giant chunk of lead. And so we took our Barley and we scanned it. So the scanner is not particularly fast. So what we did was to put the Barley, we built these foam cubes. So you get 4 Barley spikes in each of these cubes. And then when you scan it, you end up with sort of pictures that look like this. And the letters on the side are for alignment, so we can figure out which one went into which square. And so I should point out, so Michelle Quigley is the postdoc who's been doing all of the fantastic X-ray CT scanner work, and this machine is in Dan Chitwood's lab. So we have about 875-ish spikes of Barley to mess around with. And so the next process of things to deal with is, well, image processing. So we have these raw scans. We have to do some normalization of densities. You have to remove the air and the foam. The spikes off of the top, the sort of hairy stuff, causes all sorts of problems with image processing. So for the moment, we have gotten rid of it because we can't deal with it yet. And then what's left after all of this image processing are essentially these nice little clusters of the seeds that we can work with for analysis. So as our starting point, we've just been doing analysis on the seeds themselves. And again, I should point out, so this is work that's done by Erica Abmanskita, who's the grad student with Dan and I, and Timo Felders, who's a postdoc with us. So now that we have collected all of our data down, so now we have, we can individualize seeds off of these. So what we did was we segmented out the seeds. There's a bunch of outliers you have to deal with because it's real data, and that always happens. So we went through and got rid of a lot of the outliers. Outliers can be caused either by some issues with sort of the morphology of the shape itself, and so something going wrong in terms of the scanner maybe, or just in terms of misformed seeds. Also, sometimes we had some issues with separating seeds from each other, which caused some problems. But in the end, this is maybe less than 10% of our data as outliers, and we end up with about 38,000 seeds to work with. So once we have these seeds, the first thing to do is to try some traditional stuff. So what we did was we took all of our seeds and aligned them based on SVD. So if I'm going to compare these seeds, there's sort of a natural way of lining them up with each other. They all have kind of a cashew-like shape. They kind of look like maybe a ball of dough with a thumbprint in it. And so we kind of want all of those thumbprints to land in the same direction to what's going on. And so we can do some things like traditional measures based on just now we've got all these scans, and we can do it a heck of a lot faster this way than you could do with the sort of traditional biology method, which is to get an undergrad to sit there and measure lengths of all of these seeds. So from that, we can start looking at how the morphology changes over the generations of these seeds, of the different hybrids. So in this particular example, so you can see some of the traits are pretty clearly selected for. I don't know if I'm overreaching my biological assumptions here, but for instance, the area over time increases. This is regular area. This is the convex hull area. Some things are, a bit more stable, but things like height are increased over time. The maximum height is increased over time. And so a lot of the improvements can be affiliated with things like natural selection, possibly some genetic drift, which is just the random inheritance of alleles, non-random mating. In general, though, we're seeing sort of something interesting changing with the shape and structure over the generations of these different hybrids. So this is great, and we can already do this so much faster than the biologist can do by sitting there and literally pulling apart spike seeds and trying to measure them individually. But we wanted to do some more things because, for instance, one thing that does not get measured in this at all is that sort of thumbprint-like structure in the seeds. And so the goal is to be able to use some TDA kinds of things to try to tease out some of the more complex information we can find. All right, so we're going to use the Euler characteristic transform. The idea is, okay, the Euler characteristic, if you've, I don't know, wandered around Wikipedia at all, the Euler characteristic is very simple to compute for a shape. So if I give you an input shape, I have vertices, edges, and faces up to some generalization of whatever nice enough structure I'm looking at. And the Euler characteristic is just the alternating sum of these. So vertices minus edges plus faces. And it turns out for very complex mathematical reasons that as long as your underlying shape is not distinguishable homeomorphically, you end up with the same Euler characteristic. So for example, I have all of these regular solids, thank you Wikipedia, and if you sit down and compute the Euler characteristic, so just vertices minus edges plus faces, you always get 2. And that is representative of the fact that all of them, at least topologically, are indistinguishable from a sphere. So the Euler characteristic is nice because it is very easy to compute. Unlike a lot of the TDA tools that we have, this one is very, very simple. I just have to run around counting a bunch of stuff. So the Euler characteristic by itself is a bit too simple for our purposes. It doesn't really tell us enough because our seeds are also indistinguishable from spheres. So instead, what we're going to look at is what's called the Euler characteristic curve. All right, so here's the idea. If I have some sort of binary image in my example, I'm going to fix a direction. So that's going to be my omega. So in my example over here, my omega is pointing in the direction of this red line going that way. I can define a filtration function on the pixels basically by just going in that direction and increasing. So in this example, color is based on, giving me information about the filtration function. And then the Euler characteristic curve is what happens when at each of the stops in my filtration function, I look at the sublevel set, that's what's going on down here. I calculate the Euler characteristic and I get a number. And so for every A value, which is my sublevel set location, I get a number out, and my Euler characteristic curve is this integer valued time series you see on the side. So I picked a direction, but of course you can do this in many directions. So here's an example where I'm doing it in 3 different directions. If I take the same seed and do it for multiple directions, I can just kind of take each of their Euler characteristic curves for each direction and concatenate them. And I get this giant long time series. And I can use that basically as a proxy to represent my underlying shape and structure. So why is that a reasonable thing to do? There's a lovely theorem by Turner, Mukherjee, and Boyer that says that this Euler characteristic transform is actually injective on finite simplicial complexes when you embed them in 3D. So math translation, what does that mean? If I told you the Euler characteristic curve for every possible direction on the sphere, and I mean every possible, infinitely many directions, then I could have basically all the information I possibly need to know about my input data set. That's clearly not reasonable, but at least it's a mathematical end goal for why we're doing this. For what we're going to do, so let's talk about actually applying this Euler characteristic curve to barley seeds, since I think I'm already running a little late. Right, so we can compute the Euler characteristic transform, which is basically the collection of all that data. We did this on 74 different directions, 32 thresholds for just like stopping points for counting things. Stuck them all together into one long time series, and then we've got to go analyze that data. So the first thing we did was we looked at MDS projections of that data. So here's some examples where we basically took 2 different initial founders. So this is Atlas and Everest, did an MDS projection looking down to see sort of where all of these points land. And it turns out that you can separate them relatively well with just a linear SVM to sort of see the difference. So to try to quantify what we were seeing intuitively, what we did was we started looking at the sort of pairwise comparison distances based on that information. So take the SVM and then get some sort of percentage based on each of your pairs, and then you can get a number to say sort of how distinguishable they are based on this input Euler characteristic information. From here, you can do things like look at hierarchical clustering of your different types of the different founders. What we found in this particular experiment was that it turns out that we're doing a very good job of separating out the 2 and the 6 cluster, or the 2 and the 6 row Barley. So right now, if you see everything that's red on my screen, which is this plus that guy, are all our 2 rows, and then our 6 rows are all the rest of them. And so that's probably most of the shape that we're picking out, at least with this particular method. This behavior is pretty stable in terms of messing around with number of dimensions and different choices of metrics and all sorts of other options. And so this is pretty stable for a first pass. We've looked at the PCA projection of all of our data sets to see sort of where the entirety of the seed space lands. Right now, there's some interesting things we think in here based on, again, differentiating between 2 and 6 row Barley. Part of the intuition for why we think this is that essentially the 6 row Barley doesn't have as much space to grow. So you end up with more squashed kernels because they start bumping into each other. And we think that this is part of what we're picking up with all of this, this input data. All right. So I'm already a minute over, I think, right, Aris? I don't know if I'm calculating time correctly.
That's fine. You can have a couple more minutes. Okay. That's fine. Yeah.
Nope, I'm almost done. It's all right.
Yeah.
So I just want to I want to do one small advertisement, if you'll permit me.
Yeah, of course. Go ahead.
Talk about future work. So right now, this is really like a first step at using this gigantic data set. It's sort of hard to overestimate how much amazing information is stored in this literally years of genetic and morphology information. We've only used the seeds so far. Our long-term goals are to basically be able to use the whole panicle and be able to study the shape and structure of the panicle itself. There's a lot of interesting connections to be made between the genetic information and the morphology, which we have just barely scratched the surface of. And on top of that, we have access to lots of other data sets with lots of other types of plant life. So for instance, there's I believe that's a tangerine on the left. So we've been doing some analysis of the skin of the tangerine. You can use some TDA type methods to start doing things like counting rings in what are called tree cookies, which is still my favorite application just because it's called tree cookies. There's applications to interesting shape and structure in walnuts. I mean, at this point, I think the directions and possibility are limitless. Right, so advertisement, we just found out that we got fund positions. So if there, I know this is outside of the usual academic calendar time, but if there are people who are interested in applications in this direction, so this is a project that's in collaboration with Dan Chitwood, who's also on the project I just showed. Also Arjun Krishnan, who's our resident gene regulatory network expert, and Baranda Montgomery and Aman Husbands, who are the plant biologists who study what's called Arabidopsis. So if you hang out with biologists around and you know the fruit fly is sort of the standard model for lots of different things, Arabidopsis is the fruit fly of plants, completely genetically understood. And so we can do all sorts of interesting things with knockouts. Okay, so with that, I should thank my collaborators. So Eric Amezquita did the majority of the work on this project. So Michelle and Tim are also in the group with Dan and I. So Dan is the resident plant morphology expert, and, oh sorry, Dan Chitwood is the resident plant morphology expert, and Dan Koenig and Jacob Landis provided us with the data. So thank you very much for your time, and I'd be happy to answer questions.
Thank you very much, Lise. As always, pretty clear and interesting. Would you mind please unmute yourself, and let's just thank Lise and clapping for this as well. Thank you very much. Please unmute yourself if you have questions to Lise, or type in your question in the chat box, then I will happily read it out. Okay, so I can't see any questions.
Yeah, I have
Maybe
a question.
I'll go ahead with asking. Yeah, please go ahead.
Hi, yeah, thank you. So I was just wondering if you could provide some intuition or motivation behind using in particular the Euler characteristic curve in this case. Why not any of these other representations that we saw today? Thanks.
Oh yeah, sure. Okay, so a lot of the filtration, so a lot of the topological methods that you've seen are based on having some sort of filtration function defined on the space. And so depending on where your data comes from, that filtration function is going to be different and hopefully something that you can use that will encode the shape and structure. So depending on your input data, it's possible that this comes with some sort of automatic, obvious choice of a filtration function. On the other hand, the Euler characteristic transform is built for having a shape in 3D where essentially the whole idea is creating a filtration function for that. Right, so I've got this shape in 3D that I want to measure something about the structure. And so what I can do is I can basically point a direction at it, create a filtration in that direction. But then if I start trying to do that, well, great, what choice of direction makes sense for what I'm doing? And the answer is, as most things in TDA, try to not make any decisions whatsoever. And so we just use all of them at once. And so that's the reason for using this. And the original paper talks about it in terms of the persistent homology transform instead. But if you sort of dig through their proofs, you really only need the Euler characteristic transform to get all the information that we want. And then it's just a question of choosing that because it's just so much easier to compute computationally.
Thank you, Liz, and thank you for the person who asked the question. Any more questions? Yeah, I think there's a couple of minutes before I start my talk. Maybe Liz, I'll ask a question. You know, in the slide where you build your filtration for the Euler characteristics, and I think you said you will, so how many steps you use here in any direction?
Yep.
How many steps you're using?
Oh, so this 16? One, okay, so this particular picture, I think this is only 16. Let me double check because I think we've got it on a later slide. This one, yeah. So we did. We chose, and this is not 32. For any mathematical reason. This is just because it was working. So we picked 74 directions on the sphere. So this picture over here is you can basically look at. So all of the pink arrows are on or pink dots are on a sphere, and this is all the directions that we're pointing at the seed. And then we did thresholding to basically have stopping function values. So we did 32 thresholds. So that's how many values.
Yeah, so that is my question. How critical is this thresholding values? As in, so if I decided to choose, let's say 25, or I decided to choose 40, I mean, have you experimentally you chose that, or this is based on what?
Yeah, so again, this is a little bit ad hoc heuristics, but again, chosen because it was working. We've tried using fewer. For our particular example, I think I know at some point we were doing things like 16, and we were not actually seeing the structure. So the choice of thresholds is going to be directly related to the complexity of the structure you're interested in finding. So for the seeds, 32 seems completely fine, but less than that seems to not actually find everything that we want. I could definitely imagine that when we get to some of the more complex data sets, so for instance, that walnut structure, the walnut has a lot of sort of bumpy ridges on the inside. And if you're going to actually pick up all of that information, you need a lot more directions and a lot more thresholds to be able to get away with anything.
Great. Thank you.
Yep.
Any questions? There's a question from Leonid Fedorov. Pardon me if I'm not pronouncing your name very well. Shouldn't there be a finite number of directions for a given compact shape to capture all information? And he puts, yes. I guess he conjecture.
Yes, the answer definitely should be yes. I would also conjecture yes. There is a paper by Justin Curry, who's here later. I don't think he's talking about this or not, but there's been work by various groups that have proved at least finite. So the basic idea here is that the intuition behind finite is that if you have, so all of these directions are sort of spread out on a sphere. And if you have a finite simplicial complex input, that induces a stratification on your sphere. And this is a nice finite stratification. So you end up basically breaking up your sphere into pieces where intuitively your persistent homology transform doesn't change within those regions. And so yes, in theory, yes, the answer is that finitely many directions will work. The downside is I don't know what those directions are in advance. So I can't actually pick from the get-go, pick sort of a bunch of directions and sort of guess appropriately. And so that's why we're sort of sticking with this method for the moment. So yes, if there is a better answer to that, I would love to hear it. But yes, finite, yes. Controllable.
Yeah. I think, yeah. It says thanks. Okay. Yeah. Thank you very much again.